Students, so I hope you all know the topic of enzyme. You all understand what is enzyme. So I will be first starting with a simple example of enzyme. So, all right, if we talk about enzyme lipase, all of you, this is enzyme. Why? It has ASE in its ends. So ASE represents it's a enzyme, all as well as SIN. So this lipase enzymes digest lipid. Just suppose your lipase enzyme is in your stomach. Just suppose, I'm not talking about hypothetical or any other things. Just think it's in your stomach. You eat a fatty substance, I don't know whatsoever, oily fries or oily chips, whatsoever you ate, it has lipids in it. Your lipase enzyme is there. You eat an oily substance, it has two molecules of lipids. So these just suppose there are 20 lipase enzymes. There are millions, but we suppose there are only 20 lipase enzymes. These lipase enzymes digest these two, so the activity of enzyme increases. The activity of enzyme increases. So this lipase enzymes break down these lipids when there are only two. Now you eat 4. These 20 enzymes digest also these 4. So your enzyme activity increases. So what it means, as you increase the concentration, how I am increasing the concentration? From 2 I increase to 4. From 2 I increase to 4. If I drop this 2 to 1, the enzyme activity would have fallen. But I increased. So this is concentration. I increase concentration. This increase in concentration resulted in increase of enzyme activity. This is enzyme activity, EA, enzyme activity. If then I eat only 2 lipids molecules, these lipase enzymes digest it. There are 20. Before it has 4, now it has only 2. Only 2. So it decreases. There is a fall in the enzyme activity. The enzyme activity lower down. Only 2 enzymes are working. No more than that. Before 4 enzymes were working. Before that 2 enzymes were working. So the concentration increases. So there is a downfall in the graph. So we can thought that the decrease in concentration means decrease in enzyme activity, as well as increase in concentration results in increase in enzyme activity. So let's go write it all down. Increase in enzyme activity, EA enzyme activity. Increase in enzyme activity means increase in concentration. If you want to be more specific, you can say substrate concentration. So I can write FC, this means substrate concentration. And decrease in enzyme activity means decrease in substrate concentration. Because what is decreasing? Not the enzyme. Enzyme is not decreasing, but the substrates are decreasing. From 4 it dropped to 2. So first it was increasing because of 4. Now you drop it to 2. The enzyme activity also slows down. Drop, what I mean by drop? Decrease in enzyme activity. What I mean by decrease in enzyme activity? Now there are less enzymes working. Here there are 4 enzymes working. Now only 2 enzymes are working. So there is a parallel decrease. If you want to say, there is one situation also. Just suppose there are 20 enzymes. You ate 4 lipids. 4 lipids molecules. Now you give a sudden increase. You eat such a oily fry that has 20 lipid molecules. So now all the enzymes are working. You eat another 20. Let's make a graph. You eat 2 for enzyme activity, little increase. There is a little increase. You ate 20, a long increase. Then you eat another 20. That all the enzymes, these all 20 enzymes are busy digesting these 20 enzymes. As soon as they get free to digest this enzyme, you eat another 20 lipid molecules. So this means there is no increase in enzyme activity, but enzymes are still working. There is no decrease, there is no increase. Why there is no decrease or increase? Because you have 20 enzymes, all are working to digest 20 enzymes. As soon as they digest 20 lipid substrates, there are 20 more. As soon as they digest this, there are 20 more. So the enzyme activity isn't increasing, it's continuing in a straight line. Now all 20 are free. You ate another 14 lipid molecules. First it digests 20, then it digests 20, then you ate 60. So this means it will go on. The enzyme activity continues, but there is no increase. So what we can say, there is one hypothetical or even logical situation where increase in substrate concentration can lead to stationary enzyme activity with, you can add a bracket, with no increase or decrease. So we can say, by giving the conclusion, what we can say, first let's see, increase in enzyme activity means increase in concentration. More molecules, more enzyme activity, more enzymes are working. Less molecules, less enzymes are working, so decrease. Now there are more and more substrates, but there are less and less enzymes, so they are busy in digesting one lipid, another lipid you consume. So this means there is a stationary enzyme activity with no increase and no decrease. There are two situations in enzyme activity. What are the two situations? One is the increase and the decrease, second one is the stationary. I hope you all understand this topic. I tried my best to make it easy and simple and concept based top video. So thank you for watching my video. Bye guys.